So good morning, everybody. I propose that we begin now in the name of the GIGA platforms and Interface, so the technology transfer office of the University of Liège. I'm very pleased to welcome you today to the third webinar of our series GIGA platforms, key actors for your R&D. So as you probably already know, GIGA is an interdisciplinary biomedical research center, but it is also a whole ecosystem between academic research, companies, and the University Hospital of Liège. And GIGA gathers a whole range of complementary expertises and scientific equipment that are available to your academic lab or your company lab. And by the way, if you need to be connected to the right person among all the GIGA experts, Tim Bibon, who is in the audience right now, is the coordinator of the platforms, and she can guide you according to your own needs. But let's now focus on the topic of the day. Indeed, there will be two, as our webinar will be divided in two parts. First one, cell imaging, and then flow cytometry. And for both fields, we will have a presentation of the available services and equipment by Sandra Hormonez, who is the platform manager. And that will be each time followed by a testimony to illustrate with very concrete cases. But so we will spend 1 hour together. We won't have many time for questions, but anyway, don't hesitate to ask for a meeting with our speaker later on, according to your agenda. You just have to ask Aurélie in the chat conversation tool to send, to organize a meeting. You send her a private message and we will arrange that to further contact. So let's focus on cell imaging and to present the available equipment, services, and potential application fields. I have the pleasure to welcome Sandra Hormonez, who is manager of the GIGA Cell Imaging Platform. As background, Sandra is a biologist with a PhD in plant physiology. And after a few years as a researcher for the National Scientific Research Fund, she was hired by the GIGA to create the cell imaging platforms and the flow cytometry platform. So that was 14 years ago. So thank you, Sandra, for sharing your expertise with us. I would like to know that you have a very beautiful screen background. It is not coming from an art gallery. It's an image of two cells communicating together. So that's just to the point. Sandra, please, the word is yours. We are listening to you. Your screen is okay.
Okay. Perfect. So, hello, everyone. First of all, I would like to tell you that I am very, very happy and proud to present you our GIGA cell imaging and flow cytometry platform today. As Fabienne explained, my presentation will be split in two, with first a presentation of the cell imaging part, and then a talk about the flow cytometry technology. But first, I would like to introduce my team and some key figures about the activity of our core facility. The platform was created in 2007 by pooling some cell imaging and flow cytometry equipment that were present in different GIGA labs in order to have them maintained, to exploit them at the maximum of their capability, and to put them at the disposal of a very large community of researchers. This approach allowed us to realize that an ever-increasing number of users were interested in using these cell imaging and flow cytometry technologies. By meeting with the researchers on a daily basis, we were able to identify a wide variety of research requiring these technologies, and we acquired new instruments over time in order to meet the demand and to expand our equipment pool with the latest generation technologies. Today, we provide researchers with 22 flow cytometry and cell imaging instruments. And as you can see, the number of researchers and research groups has not stopped increasing since the creation of the platform and the progressive acquisition of new instruments. In 2020, nearly 300 researchers from more than 80 different research groups came to us for experiments in flow cytometry and cell imaging. 78% of the researchers come from the University of Liège, 12% from other Belgian universities, and 10% of the requests come from biotech companies. Of course, over time, the staff of our platform has been enlarged, and we are now 4 people working daily to offer you the best services in cell imaging and flow cytometry. They are myself as manager of the core facility, Rafat Stefan and Céline Van Wing, both of them technicians and experts in flow cytometry, and Alexandre Rigaud, technician expert in cell imaging and image analysis. By the way, we are looking to hear another expert in cell imaging in the near future. So if you know someone who loves cell imaging technologies and who could be interested in joining us, you can maybe give my contact information. Let's go for the imaging part of the platform. So we have several microscopes to allow you to observe your samples in bright field with sample illumination with white light, in phase contrast, showing the differences in refractive index, in DIC if the contrast is not optimal, in polarization contrast if you want to observe, for example, different types of collagen fibers, and in fluorescence microscopy that allows the detection of single molecules using antibodies coupled to fluorochromes. Concerning the fluorescence microscopes, we have in the core facility 4 different epifluorescence microscopes. First, we have an Olympus CKX41 inverted basic microscope to observe in bright field or in fluorescence. We have also an inverted all-in-one Olympus microscope with which you can make observation in bright field, phase contrast, and fluorescence. This system fits with lights and multi-well plates and offers the possibility to make stitching and overlays between the different modes. The third epifluorescence microscope, which is at your disposal, is a particular one since it is an intravital microscope that enables imaging in living animals and with which you can observe up to 4 different colors at the same time in a high-speed acquisition mode thanks to a monochrome camera. On this equipment, we have also a yellow grid laser available for photoactivation experiments. The last but not least epifluorescence microscope that is present at the platform is an inverted Zeiss fluorescence microscope, which is equipped for time-lapse, stitching, and 3D imaging, but also with an apotome module that increases the resolution of the system. This apotome module consists of a grid which is inserted into the illumination pathway and projected onto the specimen. The grid structure is superimposed in different positions, and at least 3 images of the specimen are taken and then processed in real time using the microscope software to create an optical section. So with this apotome module, you can do 3D imaging with a better resolution than with a basic fluorescence microscope. At the platform, we own also 5 different confocal microscopes that offer better sectioning than a classical fluorescence microscope. They are all equipped with different laser lines and for time-lapse, spectral detection, photoactivation, FRET experiments, and DIC observation with special lenses. Each system has its own optical characteristics and offers different advantages. We own a FV1000 from Olympus, a Leica SP5, 2 Nikon A1R, and a Zeiss LSM 880. The two last confocal microscopes are really special since they are equipped with different modules. First, the Zeiss LSM 880 is a classical confocal microscope equipped with two modules that increase the resolution of the system. These two modules are called Eriscan and ELIRA S1. Thanks to these two modules, we can offer you a system of super resolution microscopy. The Eriscan, which is a particular 32-channel detector, improves the resolution by a 1.7 factor comparing with the classical confocal system, and this with high acquisition speed and sensitivity. Moreover, the Eriscan is compatible with 3D and live imaging. The ELIRA S1 is a module of structured illumination microscopy. This module doubles the conventional confocal resolution while being compatible with popular fluorescent dyes, multiple colors experiments, and 3D imaging. Nevertheless, with this module, it's not possible to do time-lapse anymore. The last special confocal microscope that we own at the platform is a multiphoton intravital microscope that has two infrared laser lines, which allow a deep penetration of light, so 2 or 3 times deeper than confocal microscopes, and that into living tissues with less photobleaching and photodamage, and an improved signal-to-noise ratio. This multiphoton microscope has a 3D resolution identical to confocal microscopy, and moreover, allows second and third harmonics technologies to observe some fiber structures without any labeling. We are also equipped with a state-of-the-art microscope called the light sheet microscope, which is from the latest generation of imaging instruments. With this light sheet, you can acquire images of whole sample volume at subcellular resolution in a fraction of time, and you can observe organisms, tissues, and cells as they develop over days with no phototoxicity or bleaching. For thick, fixed sample, they have to be cleared before observation, and we have also strong expertise in tissue clearing. With this with the REV software, we can make very good 4D image processing, as you can see on this slide. At the platform, we also offer you the opportunity to do laser microdissection in the goal to obtain homogeneous, very pure samples from heterogeneous material, and that in bright field or in fluorescence mode. With this system, you can select regions of interest that will be cut by your laser and then collected by gravity into special tubes. We own also a live cell imaging and analysis platform placed within an incubator, and that enables automated quantification of cell behavior over time. With this system, you can do high throughput screening of different cellular processes and phenotypic assays in fluorescence and phase contrast modes. A lot of different applications can be done thanks to this high content system, like scratch assays, 3D spheroids assays, cytotoxicity experiments, and apoptosis and proliferation assays. In the near future, we will acquire a new super resolution confocal microscope with new options in order to observe organoids and spheroids up to 5 times larger than our current microscopes can achieve. We will also acquire a new IncuSite from the latest generation, allowing the observation in up to 5 fluorescence channels and offering new high content screening assay modules, especially in the field of cancer research. Alexandre and myself, we can help you to choose the best microscope to perform your acquisitions, design with you your experiments, the choice of fluorochrome, the instrument configuration and calibration. We can train you to become autonomous on the different microscopes. We can also help in writing funding applications needing cell imaging, and Alex can help you to process and analyze your images of different software. In fact, in the data room of the platform, 3 computers are available for image treatment and analysis in 2D to 5D. And we can help and train you to do a lot of things as deconvolution, stitching, segmentation, counting, extraction of fluorescent and morphology-based features, colocalization, artificial intelligence, and many, many more. But we can also perform for you cell imaging acquisition on the different instruments. We can do also for you the tissue clearing, the image processing and analysis. And we can also write scripts for image analysis to customize and automate your analysis with different softwares as Fiji, QPass, Imaris, and Arivis. To finish, we guarantee that we always try to develop new applications based on users' needs. We do our best to provide researchers with the latest generation of instruments. All our instruments are well maintained and calibrated. We act directly in case of breakdown or problems on all instruments, and we are always available to give you help or advice. I hope you enjoyed this presentation, and please don't hesitate to contact us if you have any questions or requests. Thank you.
Thank you, Sandra. We would like now to hear a concrete case to illustrate application in the field. So therefore, I'm very pleased to welcome Nicolas Jacques, who is technician at the Laboratory of Cardiology, the GIGA Cardiovascular Sciences. Nicolas is graduated from Liège High School in chemistry. He joined GIGA Research Center as a laboratory technician 8 years ago, where he has contributed to projects of excellence, such as an ERC project, a project that led to a patent and even the creation of a spin-off. Nicolas is now knowledgeable in a large range of techniques in molecular biology, microscopy, and flow cytometry. It will focus his testimony on a concrete application of the structured illumination microscope in microbiology. So Nicolas, please tell us more about this application. The word is yours.
Yes,
your screen is okay.
Okay, perfect. So
okay, thank you.
Thank you for this introduction. Already 8 years. So for me, I thought I just started this work yesterday. Anyway, it's passing very fast. So hello, everyone. And during this small presentation, I will talk about our use of this structured illumination microscopy to answer our very specific questions. So this microscopy was not only useful to take such beautiful pictures, as you can see there, this mushroom. I will just put this beautiful psychedelic mushroom, or maybe you could recognize there Italy. But we were also able to take scientifically relevant pictures that I will show right after. First, I will just introduce our project and then why we decided to go for resolution microscopy. So recently we showed, based by in vitro and in vivo model, an antibacterial activity for widely used antithrombotic drugs. You can see there a very specific pattern of growth inhibition and concentration dependent inhibition of these new antibiotics on Staphylococcus aureus bacteria. But we also developed a subcutaneous model of a bacterial infection implant, and we could also see clearly the difference in terms of growth of these bacteria when they were treated by the antibiotics. So this was only the first step. And since then, we are trying to understand how this molecule acts on bacteria. As we can see there on this small graph, there is 3 major ways to be toxic for bacteria. First, it's targeting peptidoglycan layers or membrane. This is the membrane of the bacteria. You could also be toxic by inhibition of nucleic acid like DNA or RNA, or you can also be toxic by protein synthesis inhibition. In terms of experiment, there is panel of different techniques that you can use to understand the action of a molecule by comparison of sensitive and resistant strain, genome sequencing, phenotypic analysis, target-based analysis, radiolabel precursory comparison, and so on. Our approach was, one of the techniques we used was this one. So we generated resistance mutant to be able to find how the bacteria could react, how the bacteria could adapt to the stress made by the antibiotics, and then analyzes this mutation to understand if there is point mutation on very specific target or not. We did this in parallel with other in vitro experiments, and this data suggests that the membrane could be the target of these molecules. And so this is why we decided to go for living material by microscopy analysis of this material. So since bacteria are quite small cells, the first question was, which microscope can we use? And so this is where the help of the platform members was precious for us. So not only to decide which microscope, but also to provide all the information for someone like me that at one point didn't know how to use this machine, but now it's the case. And so we decided to go for SIM technology for the structure elimination microscopy, because as you can see on this small video that Alex made for me, we decided to choose between a confocal Eriscan and SIM, and you can see there clearly that the SIM technology is clearly the best to distinguish both peptidoglycans, so staying in green in that case, and lipidic membrane staying in red. Even though with high zoom, you can see clearly that the SIM remained the best technology for us. Okay, so we could distinguish lipids and peptidoglycan layer with this technique. And following treatment, we could see some membrane defect that I highlighted in orange arrow there. So just focusing on the septum side of the bacteria, we could see clearly that the septum is affected by the antibiotics, even at low concentration, and even more at high concentration. But nothing happened on the peptidoglycan layer, so we could clearly focus on membrane and membrane integrity disruption upon treatment. But the data that we could extract was not only about the membrane integrity, because by looking more in details, we could also split this bacteria in 3 different categories based on the presence or the absence of septum and on the length of this septum. And we could divide this bacteria in these 3 points. And after treatment, we could also see differences in each phases of growth of those bacteria. Non-presence of septum means that the bacteria are after division or right before starting the division. Presence of a small septum is really the starting of division, and a whole septum just means that they will divide very soon. So, and we could clearly see these 3 categories in the non-treated one, and we could really see differences in between these categories after treatment. But also the size of the bacteria is different upon treatment. And so this is also something where the expertise of this people from the platform is very important, because for this, Alex wrote me a script that you can see a small video too of the script to analyze the size, the area, the volume of the cells. And so the only remaining things to me was just to run the script, take a coffee, and then come back to analyze the data. We could repeat this experiment with another gram-positive strain, in then Staphylococcus epidermidis, with the same answer, disruption of membrane. So we could clearly see, even at high zoom there, that there is some defect both in septum, on the membrane. There is some hole created. We can see also dead cells, debris. Yes, so same technology allow a great precision, allow us to see what we wanted to see on the membrane integrity, basically, even with high zoom. Okay, so for our very specific project, the use of this SIM technology was a real advantage. So we started with a new application for drugs. And even if we are still looking for the direct target, we know, thanks to some in vitro tests that we did also in parallel, but also with the same technology, that the membrane is affected by the drugs. And the analysis of such precise pictures also allowed us to ask even more questions, like, can we see differences between resistant strain and non-resistant strain? And we also think, just to produce an antibiotics body P, that probably with the same technology, we could see the real target, the real site of link between these antibiotics and the membrane. And so basically, the limit is just our imagination with these technologies and bacteria. So yes, so by this, I want to thank more specifically people from the platform, Alexandre and Céline, that clearly are always there for us, that help a lot on this project. Also my colleagues, and so you for your attention.
Thank you, Nicolas. Thank you for sharing this illustration with us. So let's speak now about the flow cytometry platform. So I will invite Sandra to come again and to present us this tool for analysis of complex cell populations with a large scope of applications. Sandra, please share your screen. It's okay, and we are listening to you.
Okay. Is it okay? You can hear me?
Perfect. Thank you.
Thank you. Thank you. So let's go for the second part of the platform, which is dedicated to flow cytometry. So flow cytometers instruments are also called FACS. Flow cytometry is used to count and analyze the size, shape, and properties of individual cells within an heterogeneous population of cells. Flow cytometry data is extremely quantitative and can be analyzed in depth by specific flow cytometry software program. Once the suspension of individual cells has been prepared for flow cytometry analysis from cells, tissues, or organisms, the prepared sample is placed on the FACS. A FACS contains several key components, including the sample, fluidics that move the sample into the FACS, lasers, optics, which gather the light, detector to sense the light, and a computer system to output the data into a form that can be analyzed by the researcher. Light scattering at different angles can distinguish differences in cell size. It's called the forward scatter, or FSC, and the cell internal complexity, and that is called the side scatter, or SSC. Diagrams of FSC and SSC information allow to make the distinction between different cell types according to their morphology. On the other hand, light emitted by fluorescence can identify a wide array of cell surface and cytoplasmic antigens. Therefore, flow cytometry is a powerful tool for the analysis of complex cell populations in a short period of time, and several applications can be developed by flow cytometry. We can do immunophenotyping, DNA content analysis, cell cycle analysis, cell apoptosis, cell proliferation, calcium fluxes, cell sorting. Concerning the immunophenotyping, cell subsets can be identified by labeling population-specific proteins with a fluorescent tag, usually on the cell surface, but also inside the cells. This is the method of choice for identifying cells within complex populations. For example, the analysis of immune cells in a blood sample. This application is often used by research and clinical labs. About the DNA content analysis, fluorescent dyes such as propylium iodide or PI intercalate into the DNA structure. The fluorescent signal is directly proportional to the amount of DNA and can identify gains or losses in DNA. This assay can also measure cell aneuploidy associated with chromosomal anomalies. With a flow cytometer, we can also analyze the replication states using fluorescent dyes to measure the 4 distinct phases of the cell cycle. Cell apoptosis and necrosis can be also investigated by FACS on the basis of differences in morphological, biochemical, and molecular changes occurring in the dying cell. By FACS, we can also get information about cell proliferation, since resting cells can be labeled with a cell membrane fluorescent dye, the CFSE. When the cells are activated, they divide, and half of the CFSE is passed on to each daughter cell. Cellular activation and proliferation can thus be calculated by measuring the reduction of the CFSE signal. The principle of flow cytometric calcium flux measurements is based on changes in fluorescence intensity or in emission wavelengths of a fluorophore following chelating of calcium ions. This is commonly reported as a plot of the fluorescence intensity against time. And by flow cytometry, we can also sort cells. Indeed, some flow cytometers have the ability to physically isolate cells of interest into separate collection tubes. This application is very often requested in our platform. There are 7 flow cytometers available on the platform. 5 of them are from the BD Bioscience company. So we have a Verse, a Cantor, a Fortesa, 2 FACS Aria, and we own also a Sony and a Beckman-Coulter device. 4 of them are flow cytometers analyzers. First, the most simple that we provide is a Beckman-Coulter Cytoflex. With this analyzer, you can run several applications as DNA content analysis, calcium fluxes, since it's possible to add the stimulus during the acquisition thanks to a peristaltic pump, and thus to make real-time measurements. You can also analyze cell cycle, cell apoptosis, cell proliferation, and you can do with this system simple immunophenotyping up to 4 fluorochromes excited by a unique blue laser. And a volume sensor is also available for cell counting on this instrument. We own also a FACS Cantor 2 with 3 laser lines and the possibility to do immunophenotyping up to 8 colors. You can also analyze the cell apoptosis with this system. The FACS Verse has exactly the same optical bench as the Cantor 2, but consists of a walkaway system since there are a plate loader and a tubes rack. With this system, you can also do absolute cell counting. The FACS Fortesa is the most powerful analyzer that we own at the platform, since you can do immunophenotyping up to 18 different colors with this system, equipped with 5 different laser lines and a plate loader. In the near future, we intend to acquire two new analyzers. So first, we would like to duplicate the Cytoflex with a second one equipped with a plate loader. And in the next 6 months, we will also purchase a state-of-the-art spectral detection flow cytometry system. With this, it will be possible to better manage the autofluorescence problems that are present on the classical FACS. Also, we will be able to do immunophenotyping up to 30 different colors, and therefore to optimize the detection of rare events. Since our FACS Cantor 2 is becoming older and older, we would like also to upgrade our Cytoflex with 2 more laser lines in the goal to be able to work with up to 13 colors on that system as soon as possible. Among the 7 flow cytometers available at the platform, we own 3 different cell sorters. First, we own 2 FACS area 3, exactly with the same laser lines, options, and optical bench. They are both equipped with 4 laser lines and can read up to 15 colors. They allow to solder to 4 different cell populations at the same time. The Sony MA900 is the last FACS that I wanted to introduce. It's a 4-way cell sorter, able to read up to 12 colors thanks to 4 laser lines. And we have put this Sony under a hood to be able to sort potentially contaminated samples. This system can also do cell cloning on 96 well plates. All these different FACS descriptions maybe were fast for you during this talk, but they are all available on the platform website. We own also a MacPix by Luminex system, which is a fluorescent imager, beads-based, that can perform quantitative analysis of up to 50 different analytes in a single well of 25 microliters of plasma, supernatant or serum. Céline, Rafat, and myself, we are always ready to help the researchers to choose the best FACS to perform their essays, to help them about their panels, the configuration and the calibration of the instruments. We give also training on the different flow cytometers or on the MacPix system. And also we can help in writing funding applications needing flow cytometry. About the data analysis, of course, we can also help users to analyze their flow data on different softwares available in the data room of the platform. For example, FlowJo and its different plugins is a wonderful tool that we offer in the core facility and that we can help you to use it correctly. But also, we can perform for you the follow cell sorting or cloning and your full flow or MacPix experiments from the sample to the data analysis. We guarantee that we always try to develop new applications based on users' needs. We do our best to provide researchers with the latest generation of instruments. Of course, all our instruments are well maintained and calibrated, and we act directly in case of breakdown of problems on all the FACS. And to finish, we are always available to answer your questions or to give you advice. Don't hesitate, of course, to contact us if you need information about the flow cytometry part of the core facility. Thank you.
Thank you, Sandra. Thank you for your explanation on this wide range of services and equipment. So the word is now to Thomas Marichal, who is research professor at the GIGA Institute and is head of the immunophysiology lab. After his training as a veterinary doctor, he embarked on a PhD in immunology at the University of Liège. He then obtained a Marie Curie fellowship to perform a postdoctoral training at Stanford University School of Medicine. And when he came back in Liège in 2016, he got a tenure position. Thomas and his team are investigating the immunological mechanism underlying lung homeostasis and the pathogenesis type 2 allergic asthma, with a particular interest in lung myeloid cells. So the application Thomas will speak about with us today is lung innate immunity revealed by flow cytometry and microscopy. So please, Thomas, put on your microphone, share your screen, and it's a pleasure to welcome you and listen to you.
Thank you, Fabienne, and hello everybody. So I will share my screen. Please let me know if you see it.
That's perfect.
Okay, good.
Thank you.
Well, yeah, thank you for asking me to, testimony on the services that are provided by the platform. And I have to say, the platform is very super well equipped, but above all, we are talking about a fantastic service that they can provide to the community. And Sandra is very humble in presenting that, but I really would like to emphasize that the team is super friendly, super competent. It's available sometimes at night, sometimes during the weekend when there is an issue, and they are very dedicated to their work. And I think the research we are doing could not be performed without their help and without a close cooperation with them. There is not one single day in the lab that happens without an interaction with them. So really, the work and the examples I will provide here are made available thanks to the service of this platform. Just to give a high-level picture of what we are interested in, so we like to understand how innate immune cells and how those innate immune cells that you see here on the slides, macrophage, eosinophil, dendritic cells, neutrophils, do behave in the lung tissue in the context of a steady state lung, so in the context of homeostasis, but also how they can contribute to immune-mediated disorders in the airways, like in the context of allergic asthma. So in the lab, we mainly use the mouse as a model. So we make a lot of mechanistic studies using the laboratory mouse and transgenic animals, but we always try to validate also our finding using human samples, thanks to a nice collaboration that we have with the pneumology department at the hospital. We do more and more analysis on animal species and also on ex vivo models. And we basically model in the mouse in particular some diseases like respiratory viral infections or allergic asthma when we expose mice to house dust mites, for instance. On these slides, you can have a global view of our contributions to the understanding of allergic asthma pathogenesis that we've done together with Fabrice Bureau mainly. So I will not go into the details, but you see that there have been many publications coming out, and that's for every step of the project, and I'm repeating myself here, but we really needed the help and the service of the platform, and I'm highly grateful to them. So I will try to just give you some illustrations of how we work with the platform for our research. And I will start with the fluorescence activated cell sorting. So we look at cells that populate the lung tissue that sometimes are very complex to identify, that sometimes are very rare. But thanks to the FACS and thanks to the FACS area 3 in particular, we are able to, from lung single cell suspensions, to isolate and sort with a very high purity and high variability and without any substantial contamination, to sort those cells. And once we have collected them, we can do several things. We can either re-inject them into recipient animals to understand exactly what they do in vivo. We can also do some ex vivo assays, ex vivo cultures, or ex vivo co-cultures with other cells, or we can obtain very good quality material to do some high throughput analysis like single cell RNA-seq or bulk RNA-seq. And to mention just the first application here, in this paper, for instance, we were interested in understanding the function of particular neutrophil subsets. And so we were able to use these FACS to sort high quality and high purity neutrophil subsets and then re-inject them into recipient animals with the appropriate controls. And we were able to show that one particular neutrophil subset that was recruited after a low endotoxin exposure was basically having a big role in the initiation of allergic asthma. So we can do these adaptive transfers with neutrophils. We do also some other adaptive transfers with monocytes, macrophage, and this is only possible when we use this with this FACS technology. We can also sort the cells and then try to understand what they do ex vivo. So we, as an example here, we sorted 2 different subsets of macrophage population from the lungs of mice. And then we put this macrophage into culture overnight with or without a stimulation with endotoxin. And then we were able to make a proteome profiling to identify which are the cytokines and chemokines that these cells are secreting, and to so identify also functional differences between these very rare and subset complex subsets of cells. And then over the last years, we've started implementing single cell technologies, single cell RNA-seq, single cell ATAC-seq as well. So these technologies, they look appealing to a lot of people, but one has to keep in mind that if you don't have good quality material at the beginning, then all your data you will generate can just be thrown into a trash if the cells are not in a good state initially. And so the processing of the cells and then the way you sort or isolate them is very, very important to be able to correctly interpret your data. And I can tell you already today, because we have replicated the experiments in different animal facilities, in different institutes, I can tell you that we here at the GIGA, and thanks to the platform, are able to really collect high quality material that you can then further use to, in this case, for instance, study and investigate the transcriptome of single cells through the RNA, the single cell RNA sequencing. So first we work with the imaging platform, and then we work with the genomic platform, and we use the 10x genomics technology to do single cell RNA sequencing as it's shown here. So this was for the sorting of the cells, which is very important in our field. And obviously, when we look at our cells of interest, we also would like to characterize them in depth, and so we can do some immunophenotyping, multicolor immunophenotyping with the FACS-Cantor or with the Fortessa. Currently, we use 12 to 14 colors on the Fortessa, and I'm very excited that we will be able soon to go up to 30 colors with maybe the Cytec Aurora. Again, here this is something we use every day in the lab. We take lungs, we digest them, we have single cells, and then we stain them with some either extracellular or intracellular markers. And this allows us to identify them, phenotype them, and also have some insights about their function. This is an example of the gating strategies we use. And then recently, we also use different softwares. And again, Céline from the platform is very helpful in helping us so that we can basically have a view of all the cells we have analyzed on either TSNE or UMAP, a bit the same way as you would see it for single cell RNA-seq, except that here we have a UMAP or a TSNE that is a representation of our 14 markers and not of the hundreds of genes as you would have in single cell RNA-seq. And when you have this UMAP, you see all your cells, and then you can interrogate them for the expression of the different markers you are using. So here you see that you have like 2 clusters that express high level of LY6E, for instance, and so on and so forth. So this is very routine work in our lab, and this is and Céline is very, and Rafat previously, and he will come back, and Céline are very helpful for that. So this was for the flow cytometry part, and well, it's nice to be able to isolate the cells and look at them through a cytometer, but it's also very important to see the cell in the tissue. And more in my research, I'm more interested in tissue biology. So we need to see the cells, we need to see how they can interact with their microenvironment. And for that, we need to use microscopy. And the microscopy equipment is also fantastic. I am here showing you an example of high-resolution confocal microscopy we've used recently during the first wave of the COVID-19 crisis. We collaborated with clinicians, we had access to lung biopsies, and we wanted to investigate whether we could identify some particular functions of neutrophils, which are reflected by their ability to release neutrophil extracellular traps. We were interested to see if we could detect that in the lungs of severe COVID-19 patients, because we think that it can contribute to many features of severe COVID-19 pathology. And we really had to go fast because we know it was a very hot topic, and again the platform was working together with us, and Alex was like very committed to this work with us to not only analyze acquire the images. As you can see here, we actually detected very nice neutrophil extracellular traps in the lungs of those patients, in the vessels, in the airways, in the interstitium. And together with Alex, we developed an algorithm that allowed us to quantify on the sections, on the Z stack actually, the volume of those net structures. So it was not only about seeing those nets, but it was also about quantifying their volume so that we can have an objective view of what's going on in the tissue. Then Sandra mentioned the Eriscan mode. So this is very interesting because you can really go to a very high resolution. You are able to see subcellular components even in a lung. I mean, it is paraffin embedded lung section here, and you see those neutrophils with the myeloperoxidase in green that is located in the granules. So you can identify activated neutrophils. You can also identify this orange cloud, which are the actual neutrophil extracellular traps. So beautiful pictures, but also very informative. And it's nice to have sections, but it's even more nice if you can have a global picture of your whole lung. And again, we worked together with the platform to develop a clearing technique for the lung, which was not so easy. But as of today, we are able to have an optimized clearing technique, and we can do stainings on those whole lung, and you can so have here stainings on the whole lung, and you can then zoom in and have information about the localization of your cells and what they could actually do. So this was my my illustration of the services provided by the platform, and you know, I hope I could convince you that this is really a wonderful team to work with. So thanks a lot.
Thank you, Thomas. You were really convincing. Thank you for your explanation and your kind recommendation. I've noted super friendly and super competent. So that's what you said. So that's a reminder to all of you in the audience that Sandra and her team are at your disposal. Sandra would be very pleased to meet you and discuss with you about your project. So you understood that. Thank you. So it's about time to close our public sessions, to respect our timetable and your agenda. So a special thank again to our keynote speaker for their availability, for the relevant information they give us and they shared with us today. Thank you also to my colleagues at the back office having taken part to this organization. So there is Aurélie, Karine, Annick, and Florence. Thank you to you. for attending today. So I hope that there will be a further connection with the platform. I remind you that our next webinar will take place on the 6th of May, and the topic will be genomics. The session has been recorded, so Aurélie will send you a link so that you will be able to review it or to share it. So thank you again. I wish you a nice afternoon and success for your own project. Bye-bye.